welcome. I am about halfway through my PhD with Anthony Vernon at King's College London, and I'm funded by the NC3Rs. And my overall kind of running title of my PhD at the moment is using human stem cell derived microglia and neuroprogenitor cells to model the effect of IL-6 on early neurodevelopment. So taking a real step into the molecular world, if that's okay with you guys. And what I'm going to present to you today is certainly within the realms of maternal immune activation, which has been obviously very well introduced already, thank you, where we know that mothers who experience some kind of immunological activation, whether it's from stress or whether it's from an infection, autoimmune diseases, and even genetic predispositions, that's priming a risk for neurodevelopmental disorder in the offspring. And one particular factor of this is certainly increased IL-6, which we're seeing and is clearly very important. And obviously, as we've seen already, it's a predictor for adjusted brain volumes in the amygdala, and also how well offspring are performing in cognitive functions. So there's certainly something happening in terms of maternal IL-6 levels and the offspring's risk for neurodevelopmental disorders. But as we understand it at the moment, the actual molecular causality for this isn't super well understood, certainly within a human relevance. So a lot of the work that's been done already has been done in rodent and non-human primates. And even this paper, which came out more than a year ago now, showed that DAMs, so pregnant mothers that had been exposed to poly-IC, which is a viral mimic, to stimulate that immune response in the mothers, actually had increased IL-6 in the liver, placenta, and also in fetal primary microglia. So it may not necessarily be that the IL-6 is being transmitted all the way through to the fetal brain, but there's certainly an increase of IL-6 in these specific tissues, which is important. So we're kind of understanding it to be a sensor transducer and effector of maternal immune activation risk for neurodevelopmental disorder. But we see these differences in rodent models and how their brains develop in comparison to human physiology. And it's kind of argued that some animal models can be quite wasteful. So there's certainly scope to model the risk of maternal immune activation mediated by IL-6 in a human relevant model that reduces animal waste. So we would argue that using human induced pluripotent stem cells could be a great model to try and understand this molecular causality, given that the movement from pluripotency through to specific cell types is relevant to development. And also you have a massive benefit of being able to derive many different cell types with the same genetic background, and it reduces animal use. So what I'm going to show to you today over the last 2 years is work that I've done to try and understand how IL-6 exposure to both human-induced pluripotent stem cell-derived neuroprogenitor cells and microglia can cause changes in their molecular phenotypes with relevance to neurodevelopmental diseases. So the methods that I've done so far are we've taken iPSCs and we've derived microglia-like cells and neuroprogenitor cells separately in monoculture first, so that we can stimulate them with IL-6 and then look at their response to IL-6 individually first in qPCR, immunoblotting, RNAseq, and live imaging. And this has been done in 3 healthy male donors, each with 3 individual technical repeat clones per donor. So the first thing that we sought to do was to see if these cells in monoculture could actually respond to not only IL-6, but also any other cytokines that they might be producing as a secondary to the response of IL-6. So we measured the receptor expression at a transcript level for things like interferon gamma, TNF-alpha, IL-17, TLR4, and the IL-6 machinery itself. And you can see that through the differentiation period of these cell types, certainly in microglia, these receptors were increasing. And similarly in neuroprogenitor cells, apart from the IL-6 receptor machinery, so this is something that I'd like to maybe pull your attention to, with the microglia-like cells, we see that the IL-6 receptors are increasing through differentiation, but actually this receptor, which is essential for receiving the IL-6 stimulus in neuroprogenitor cells, is lost upon neuralization. So that was a first kind of indication of maybe these neuroprogenitor cells are not able to respond to IL-6 in monoculture. And this is important because it's also an indication that the neuroprogenitor cells are receiving the IL-6 stimulus via a different pathway of signaling. So IL-6 works via either classical signaling, where cells will express this GP130 receptor and the IL-6 receptor alpha to bind to IL-6, which then forms a complex and induces a downstream response to IL-6. But if these cells do not express this this IL-6 receptor alpha, they're able to receive it in solution from an extracellular matrix via cleavage of this receptor from cells that do express this receptor. So this receptor will be cleaved on the cell surface membrane, and then that will bind IL-6, which will then be able to form a complex with this GP130. So if you go back to the differences in the neuroprogenitor cells and microglia-like cells that we were seeing, the microglia are able to express both of these subunits that were required for IL-6 signaling, but the neuroprogenitor cells aren't. So it's likely that these cells actually receive IL-6 via trans signaling. And this matches up to some brain RNA-seq data that we found, where we're seeing that the IL-6 ST, or this GP130, is happily expressed in microglia and neurons in all different cell types where they're able to receive that IL-6 signal via trans signaling, but the IL-6 receptor itself is actually only really expressed in microglia in the brain. So the first indication that these cells might not be responding in monoculture. So next we wanted to check whether these cells were these receptors were functional. And we did that by stimulating microglia-like cells for 3 hours and 24 hours with IL-6, and also neuroprogenitor cells for 3 hours. And what we found was that the microglia-like cells increased transcripts of IL-6 itself as a positive feedback loop, JMJD3, which is a downstream transcription factor of IL-6, and IL-10 itself as well. And after 24 hours, this response was certainly resolved at a transcript level. But after 3 hours of stimulation in the neuroprogenitor cells, we weren't seeing a similar response. So we weren't seeing any changes in any of these, the IL-6, TNF-alpha, or JMJD3 downstream transcripts. So perhaps they weren't responding, but this could also be specific to the genes that we're obviously measuring, right? So we wanted to look at it at a protein level as well. So we checked for STAT3 phosphorylation, which is I guess a key sign of activation of the IL-6 pathway. And we could see that in the microglia cells, the STAT3 was being phosphorylated after 15 and 30 minutes of stimulation with the IL-6, which is pretty fast. But nothing was happening with the neuroprogenitor cells. So we were pretty certain that these neuroprogenitor cells were just not able to respond to IL-6 in monoculture. So we and I guess as a final thing, just to check with the microglia, we sought to try and find the minimal dose that we could use that would be physiologically relevant to those seen in maternal serum. And actually, because previously we'd been using a dose that is 100 nanograms per ml, which is relatively high in comparison to what you see in the maternal serum, which is closer to a picogram per ml. But actually, with these transcripts that we were looking at, and these two colors are just two different male donor lines, we could see that after 100 nanograms per ml, the response actually dropped off pretty quickly after 3 hours of stimulation. So we continued to measure the response from just the microglia in monoculture to 100 nanograms of IL-6, given that we knew that that would bring us a response. So just to summarize so far, we can see that the neuroprogenitor cells are losing the expression of an essential key receptor for IL-6. And the microglia are able to respond to IL-6 in monoculture, but the neuroprogenitor cells aren't. So we then used 3-hour treated microglia samples for RNAseq to see more of a kind of a larger bulk response from the microglia. So just to run through it very quickly, biological samples were prepped, sequencing reads were sent for PASQ files, and then we aligned via the STAR method, counted with R subreads. So all of this analysis was then done in R, and we used DC2 for differential expression analysis, and then downstream gene ontology analysis. And just with this very quick PCA, you can see that the samples were clustering by treatment, although two of the donors seemed to respond in a fairly similar way. And there was an increase of 156 genes that passed 5% FDR, and 22 genes were decreased in expression after IL-6 expression. Stimulation. So basically, the genes that really caught our eye that were being increased by this IL-6 stimulation were things like REL, which is a subunit of the NF-kappa B complex, heat shock proteins, and also the oxytocin receptor, which could be of interest for future work. We took the select 156 genes that had gone up in the IL-6 stimulation, and through gene ontology analysis, we can see that these microglia-like cells are certainly responding in a way that you would expect from a myeloid cell. So we're seeing response to cytokine, we're seeing production of interferon gamma in response to interferon gamma, which obviously could then have a secondary effect on the downstream neuroprogenitor cells that they may be in contact with. And finally, of interest, this group of genes that were upregulated in our analysis actually significantly overlapped with a gene set that was increased in schizophrenia post-mortem tissue from humans, and also trended to increase with those that were increased in autism patients as well. So this kind of showed us that the cell model that we're beginning to create actually might hold some relevance to real life settings in human physiology. And finally, we checked for microglia motility after IL-6 treatment. And this was done in 2 healthy lines. We're beginning to collect the 3rd healthy line at the moment. And generally what we're seeing is a decrease in the roundness of the cells and an increase in the area of the cells. So that's kind of maybe indicating an increased surveillance state that these cells are gaining upon IL-6 exposure. But so far, no changes in speed or distance, either at the nuclear level or at the cytoplasmic level. But that might change once we collect the 3rd replicate. So to conclude, we're seeing that the neuroprogenitor cells are losing the expression of this IL-6 receptor. The microglia cells do respond in monoculture, but not the neuroprogenitor cells. So all of this to say that if we are going to continue to make this model of IL-6 exposure and showing risk for increased neurodevelopmental disorder, we need to create a neuron microglia co-culture to try and understand this at a molecular level. So the outstanding questions that I'm going to aim to test towards the end of my PhD is to create a trans-well shared media system where we're going to grow the neuroprogenitor cells on the bottom of a well, and then we have a mesh insert where we grow the microglia-like cells on top of the well so that they share media, so they're able to share any cytokines that are being produced by the response of the microglia, which will then be imposed onto the neuroprogenitor cells. And they're also able to share that integral IL-6 receptor for the neuroprogenitor cells to also maybe respond. And then quite interestingly then bring in the aspect of genetic risk. So the 22q11 deletion is something that increases risk for schizophrenia. And we have access to these patient lines where they have this 22q deletion. So we would grow healthy male donors against 22q deletion patient lines and compare how these co-cultures respond to IL-6 and whether the incorporation of that risk also then changes how the neural progenitor cells develop. So thank you very much for listening. And also thanks so much to my two amazing supervisors, Tony and Deepak, and to everyone else in the lab that's helped with my work so far. And I'd be really happy to take any questions.
Thank you so much. This was really a very, very interesting talk, and I'm sure there are a lot of questions. Unfortunately, I don't think I can take any because of the interest of time, we have to move on. Very sorry about that, because I'm sure there are a lot. So please, please contact each other and start the discussion. But yeah, thank you so much for this talk, but we do have to move on.